Hello, everybody. Welcome back to biology class. So as I kind of mentioned in that introduction to distance learning video, what I'll be doing here is occasionally posting sections of PowerPoint slides, which is a little bit of narration over them. So I'm not going to post, very long slideshows. I'm not going to sit here and talk for 45 minutes. Nobody wants that, including me. So I'll just have these series of videos posted every now and then, just going over maybe 10 to 15 minutes at a time, just some of the material that we're covering. So we were in the middle of protein synthesis, and I did assign a online quiz for you to take. We were right there at the end. I was going to give you that quiz on Friday, but unfortunately, of course, we were not at school. So I went ahead and I put that online. The link and the code you need to get into that quiz are posted in the assignment folder for Friday. But in any case, here we are starting Unit 9 with genetics. So as we start off this unit, however, I kind of want to give you just some things to think about. First of all, are DNA mutations always bad? Is there such a thing as a good DNA mutation? And also, what do you think would happen to a protein if a DNA sequence was mutated? So remember that proteins are, of course, made out of what? Amino acids, correct? Yep, amino acids. And that amino acid sequence that makes up the protein is, of course, determined by a particular section of DNA, the code that's in that section. So if we end up with a different code than what it's supposed to be, then of course we're going to end up with a different amino acid sequence. And so if that happens, we could end up with either a non-functioning protein or a protein that is not acting in a way that it should. So let's first start just by talking about mutations in general. So mutations themselves, certainly not there to give anyone superpowers like they show in a lot of movies. Really, mutations are just these changes in the genetic information encoded within your genes. So for the most part, DNA is actually a very highly stable molecule. It doesn't really suffer that many mutations. If you remember way back to Unit 7, I think I mentioned that these mutations happen maybe about once every 100 billion times there might be one mutation. So for the vast majority of times, the DNA is replicating and building new strands. It's building the same molecule over and over and over. It's just that every now and then there is that mistake, and those mistakes sometimes can lead to mutation. Not always, but sometimes they do. So if we want to talk about different types of mutations, we could start here. Of course, if we were to look at the very far left over here, left-hand side, you can see at the DNA level, the code is supposed to be TTC, so thymine, cytosine. And with that code, the corresponding messenger RNA would be AAG, adenine, guanine. Because remember that T always binds with A, and that C binds with G. So the messenger RNA sequence AAG leads to the building of the amino acid lysine. However, we could have a mutation in this code at the DNA level, which would result in a different messenger RNA being made. If a different messenger RNA is made, there's a chance that a different amino acid is made, or is acquired, I could say, by the transfer RNA. So let's look at some situations where this might happen. There is such a thing as a silent mutation. So if we can see here our original DNA sequence with TTC, we now have a DNA sequence of TTT. That's going to result in a messenger RNA of AAA being coded for. AAA, however, codes for lysine. That's the exact same amino acid that was supposed to have been gotten by AAG. So these two different messenger RNA codons actually end up coding for the same amino acid. And so therefore, if it's getting the same amino acid that it was going to get anyway, then it's not going to affect the protein. And so therefore, it is just known as a silent mutation. There's not going to be any effect on the protein that's built. On the other hand, however, we could have what's called a nonsense mutation. In a nonsense mutation, what happens is the protein that's coded for ends up instead coding for a stop codon. If you remember back to the codon charts, codon chart, excuse me, remember we had one start codon and then 3 stop codons. So with a nonsense mutation, we end up coding for the polypeptide to basically stop building. And so therefore, we could end up with a half-formed protein. Perhaps it would have just started on its trek to building a protein, and now it's already stopped. And so therefore, we'll end up with a pretty much a non-functioning protein if that happens. So we definitely do not want any nonsense mutations. We don't want these proteins to stop being made in the middle of their production, or at the beginning or even at the end of their production, because it could mean that that protein does not work the way it's supposed to. So if we go over here to missense mutation, don't worry about conservative versus non-conservative. What you really need to know here is that a missense mutation just results in an entirely different amino acid being made. So where we wanted the sequence TTC to help create create the amino acid lysine, instead we end up with TCC, which is instead going to make arginine. Or instead of TTC, we end up with TGC. TGC is going to code for threonine. So neither arginine nor threonine are lysine, and therefore we end up with an entirely different amino acid than what we were supposed to have had. That could very well affect the shape or the functionality of the protein, and so we might have either a non-functioning protein or a protein that does not work the way it's supposed to. So silent, nonsense, and missense, 3 different types of point mutations that could occur. Other than those 3, we also have what are called either substitution, insertion, or deletion errors. In a substitution error, we end up with, on the DNA sequence, we end up with a base substituting for the one that was supposed to have been there. So if we were to look up here at our original DNA sequence, you can see right here this 3rd codon was supposed to code for cytosine, thymine, thymine, CTT. But instead of this cytosine, we end up with another thymine. So we end up with thymine, 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 TTT. The cytosine has been substituted for by thymine, so it's a substitution error. When that happens, however, instead of making the amino acid glutamine with the corresponding messenger RNA code GAA, we end up instead making lysine with the corresponding messenger RNA AAA. So that would be a substitution error. We could, however, also have an insertion error occur. In that case, we've actually inserted an entirely new base in front of the code that was supposed to be there. So we see we're supposed to have CTT, but instead we have CCT, and the T that was on this codon here has instead been pushed to the right to go onto this codon instead. So therefore, we've inserted an entirely new base, another cytosine there. It's going to result in the formation of an entirely different mRNA codon, GGA. And that codon is going to give us an entirely different amino acid than what we were supposed to have had. And because the entire code has been pushed to the right by one base, we're going to end up with a different amino acid down here as well for the next codon. And so just because of the insertion of one base, it could mess up this entire polypeptide sequence. Deletion errors, kind of like the opposite of an insertion error, of course. We had a base that was supposed to have been right here, but instead it has been taken out, and the entire sequence, therefore, is moved to the left by one base. So now we're going to end up with 3 different amino acids than what we should have, because all the corresponding codons have also had to move down to the left by one base. So we should have had an adenine right here, but instead it has been deleted, and we end up starting our codon with a thymine. That's going to give us the sequence thymine, guanine, cytosine, instead of adenine, thymine, guanine. And so our corresponding messenger RNA is therefore different, and our resulting amino acid would also be different than what is expected. So these are just 3 other types of errors that are possible through mutations. So mutations don't always exist just at the basically DNA or transcription level. They could also happen at the translation level. So remember, we're taking our codon, our sequence, and basically turning that into a corresponding amino acid. So ACG generally codes for threonine. However, not always, this happens rarely, but not always will it actually read in the sequence it's supposed to read, or translate in the sequence it's supposed to translate. And so therefore, instead of starting with this adenine, the codon that's read inside the ribosome is CGA. Remember, it's supposed to be ACG, but it's basically dropped this A and started reading from this C, so it's reading the sequence CGA. So that's going to build arginine as our amino acid. And instead of AGG, we're going to have GGA. So it's going to be an entirely different amino acid. So this +1 frame shift translation error, what it's referred to as, is going to end up building an entirely different protein. If you look at all these bases, we're supposed to have threonine, arginine, threonine, alanine, tyrine. Now we're going to have arginine, glycine, leucine, and so on. So we're going to end up with an entirely different protein than what we should have had. And so therefore, we're going to end up with either a non-functioning protein or a protein that we really don't want. The opposite could also happen, where we end up dropping a base on the right-hand side, where it starts reading over basically, it'll start with GAG, so it's starting here, it's reading GAG, and it's actually shifted over to, see, it drops the AC, never even reads them. And so it starts with GAG, building glutamine, and once again, the sequence starts and is building lots of amino acids that we did not want. So it's going to build a protein that was not originally intended. So these errors can happen both at the transcription and translation levels. So just some examples here of a missense mutation. Dwarfism results from that. Muscular dystrophy comes from a nonsense mutation. Cystic fibrosis, the disease that we're raising money for through SuperDance, is actually the result of a deletion error. And then Crohn's disease can come through due to an insertion error. So these are just some examples. There are definitely a lot more. The question always arises, well, what exactly causes this in the first place? What causes these mutations to occur? And what can occur is that exposure to certain mutagens can cause DNA to basically mutate, can cause DNA to perhaps read the wrong sequence, and cause these sorts of either even insertion, deletion, or substitution errors. So basically, thinking about like a prolonged exposure to sunlight, UV radiation is going to cause the cells inside your skin to basically undergo certain mutations, which could lead to uncontrolled growth and result in a tumor or cancer. So there are these certain chemicals and radiation effects that can actually damage DNA and result in these particular things happening. So this is just some, beginning background knowledge about what exactly mutations are, how they affect DNA. From here, we're going to go directly pretty much into genetics, where we'll start the next sequence with talking about vocabulary and the history of our understanding of genetics and inheritance. But for now, we'll go ahead and leave it here, and we'll pick up again next time. Hope you have a great day. Thank you.